When Mendel came up with that idea, his idea of P generation, F1, F2s, and he was looking at the characters and the traits associated with the characters. Remember, character, flower color, trait, purple or white. To try to explain the data he came up with, the numbers, the patterns in the numbers, he developed some theories, some ideas that, almost 2 centuries later, we're still going, yeah, that works. He had 4 concepts, 4 ideas. The first concept, his first idea, is that there are alternate versions of genes that account for variation in inherited characters. What does that mean? Well, that means that the character, flower color, had to have some different versions of that character in however the blueprint, the instructions are saved. Hence, purple or white, tall or short. These characters from an organism are going to come from, 2 versions in every organism, 1 from each parent. Nowadays, we rewrite that to say alleles. Every gene's going to have 2 alleles. Each of those 2, one of them you're going to get from mommy, one of them you're going to get from daddy. You look here at this picture, this artist rendition of one of the pea plant chromosomes. Somewhere on the chromosome is going to be the gene that controls flower color. We now know all organisms are plants, animals, going to be diploid, at least. And there, say this is chromosome 6. On chromosome 6, there will be a gene for flower color. That gene can come in one of 2 forms. The gene, the allele, the flavor, is going to be either purple or white. Now remember, in an earlier lecture, when I brought up the word allele, I told you to think ice cream. Everybody knows ice cream. And when I say ice cream, when everybody thinks ice cream, pretty much everybody thinks of almost everybody thinks of a different flavor. The gene is ice cream, and what flavor you think about is the allele. Here, the gene is for flower color. The allele, or the flavor, can either be purple or white. In modern science, in modern genetics, we know that the gene is nothing more than a blueprint. Blueprint that's spelled out in A, G, C's, and T's. That blueprint the blueprint will describe how to build an enzyme. An enzyme that will make purple pigment. If that allele, that flavor is present in one of the 2 genes on how to make the enzyme, purple pigment will be made. Hence, you will have a purple flower. On the other side of that coin, if the allele is for white flower, well, turns out, as you see here in this figure, that is what they refer to as a mutated gene. That mutated gene is incorrect. The blueprint is messed up. The blueprint does not contain the instructions on how to make a working enzyme. No working enzyme, because it has the wrong shape, no function, means that no purple pigment can be made based on the instructions of this allele. So we now know if the two alleles that are present, if one of them has what's referred as the dominant, here with flower color, it's purple. The other one can be white, but the first, the dominant, still calls for the making of enough enzyme to make enough purple pigment for the flower to be purple. But if both alleles are mutated, thus the white allele, neither one will have the instructions on how to make a functioning enzyme. No functioning enzyme, no purple pigment will be made, thus the flower will be white. So as long as at least one of the alleles has the instructions to make a functioning enzyme, that is considered the dominant.